and share the screen. Are there any questions before we get started? All right, if there's no questions, let's continue. We talked about what... Did we talk about this slide? I don't think we've talked about this slide yet. Okay, I've got it marked that we did this one, but I think we were going to that one and I hadn't talked about it yet. Microbiology and microorganisms. Make sure I'm recording. There it is. All right. So what comes under the umbrella of a microorganism? What is a microorganism or a microbe? The bacteria, archaea, they are obviously microorganisms. But viruses and prions are also microbes. You could argue that viruses and prions are not organisms because they don't have cells, but we do study them in microbiology, so we do call them a microbe. And a microorganism is used synonymously with a microbe. But remember that viruses and prions are not cells, so they're not organisms. Certain of the eukarya are also microorganisms. All of the protozoa, the molds and the yeasts, some of the algae. If the algae is single-celled, or even if it's colonial but it's small, we consider it a microorganism. Now, the algae that washes up on the ocean beaches, like the kelp, that would not be a microorganism. So it's only some of the algae are microorganism. And this may surprise you. The helminths, meaning what most people call worms, are considered a microorganism. But most worms aren't microscopic, right? So why are we considering the helminth a microorganism?
Is it because of in certain life cycles you can study them under a microscope?
Yes. The helminths at various stages of their life cycle are microscopic. In fact, the infective stage of a helminth, at least I should say most helminths, I guess there are a few exceptions where they actually burrow into your skin, but most of them, the infective stage is a single-celled fertilized egg, which is usually consumed, and that is the infective stage. So some of the stages of the worms are microscopic, even though most worms are, as an adult, not microscopic. Like the beef tapeworm, I think it's 6 meters long. It's definitely not microscopic, but it starts life as a single-celled fertilized egg, which is then consumed. So some of the life stages are microscopic. Another reason why microbiologists study the helminths is they study organisms which cause disease. And the helminths cause disease in humans. So the microbiologists study them. Anyways, the helminths, some of their life cycle, they are microscopic. So they are considered microorganisms. And they do fall under microbiology. Now, when we're talking about the helminths, you should understand that the worms are more than one phyla, so they're not all the same. Many of the worms are parasitic, some of the flatworms, some of the roundworms, but not all of them are parasites, meaning most of the worms do not cause human disease. In fact, the annelids, which you can find an earthworm in, if you remember what an earthworm is, they're the annelids. None of them cause human disease. It's only the flatworms and the roundworms. Some of them cause disease, but not all of them. So only some of them are parasitic. Okay? Any question about that? Anyways, that is what falls under the umbrella of a microorganism. Let's talk a little bit more about viruses. We did mention that they are a microbe. They are acellular, meaning they lack cells. They do contain a genome, but their genome is different in that it could be DNA, but it could be RNA. And it's never both. It's either one, DNA, or RNA. It's never both. And that is the core molecule for a virus. The core is surrounded by a protein coat. So the DNA or the RNA genome is surrounded by a protein coat. The protein coat may be enclosed by a lipid envelope, but not all viruses have a lipid envelope. Only some have a lipid envelope. And when do viruses replicate? A virus replicates only inside its host cell. So viruses only can replicate when they've infected their host cell. Viruses are not part of the three-domain system, and that's because they're not cellular. In fact, you could argue whether they're alive or not, but we're not going to go there. All right, any question about viruses? We will talk a lot more about viruses. About 2/3 of the way through the term. We'll have a whole lesson on viruses. But I just wanted to introduce them now. So let's return to the three domains of cellular organism, because only the cellular organisms fall within the three domains. And then let's talk a little bit about each of them. Sorry, I wanted to get my face out of the picture while I was scratching. The three domains are bacteria, archaea, and the eukarya. And let's talk a little bit about how they're similar and how they're different. For the cell type, the bacteria are prokaryotic cells. The archaea are also prokaryotic cells. And that differs from the eukarya, which are eukaryotic cells. When we look at the chromosome, the bacteria generally has one chromosome, and it's circular. When I say circular, that means it begins where it ends, so it's in a circle. The bacteria have no histone proteins or introns, and we'll talk a little bit about them more. A histone is a protein in eukaryotes, and you find it on the chromosome. The bacteria have no histones or introns. The archaea are similar in they have one chromosome, and that chromosome is generally circular, but it differs from the bacteria in that you can find histones and introns, I don't know what happened to the spacing there, in the DNA of the archaea, meaning on the chromosome of the archaea. The eukaryotes generally have many chromosomes. Apparently there's one or two species that have one, so one to many. Most of them have many. Humans have 46 chromosomes, and they are a linear chromosome. Like, for example, in the X, it is a starting position and an ending position. So it's a linear chromosome. And our chromosomes have histone proteins on them, and we have introns in the DNA. And we'll talk more about both histones and introns later. When we look at the ribosomes, the bacteria and the archaea have ribosomes which are smaller than the eukarya. The bacteria and the archaea have ribosomes of 70S size. The 70S is the position where the ribosome sediments in an ultracentrifuge gradient. And if you don't know what that is, I don't think you really need to know. But the biochemists take the ribosomes and then put them in an ultracentrifuge gradient, and the smaller ribosomes sediment at the 70S position. The larger ribosomes, which are found in eukaryotes, sediment at the 80S position in the ultracentrifuge gradient. The important thing is 80S is just bigger than 70S. When we look at the cell wall of the bacteria, we find the molecule peptidoglycan in the cell wall. When we look at the cell walls of archaea and eukarya, there is no peptidoglycan. So the eukaryotes in the cells, there is no peptidoglycan. And that's really how the bacteria differ from all the other domains. All right, any other questions about comparing the three domains? Let's compare looking at cellular microbes and acellular microbes. So the cellular microbes, we're now lumping both the prokaryotes and the eukaryotes, meaning all three of the domains here, comparing them to viruses and prions. And you know, we should have a line up here. It's like there's a line missing. The cell type of the cellular organisms are, of course, prokaryotes and eukaryotes. The cells have a plasma membrane. You can find cytoplasm and chromosomes and enzymes in the cell. And the cell is pretty complex. A lot of things are going on in a cell, and they have a lot of molecules in them. So we're just going to say, simply put, the cells are complex. Viruses, on the other hand, are acellular. They do have a genome, which you could call a chromosome, and it's inside a protein sac, the protein coat. But there is no plasma membrane, there is no cytoplasm, and viruses are pretty simple. The simplest viruses only have two molecules, like DNA and one protein. Of course, most of them have more than that, but Like I said, the simplest ones are really simple. And even the most complex virus is simple compared to a cell. Prions are even simpler. They are acellular. They are a single protein molecule. There is no genetic material, no plasma membrane, so they're really simple. And we'll talk about prions when we're talking about viruses, so that'll be later in the term. Any question about that so far? All right. If we look at the genome, and I should use that word rather than chromosome, the cells have a genome that's made up of DNA. When we look at the genome of the viruses, the genome can be DNA or RNA, but it is never both. Prions have no genome. All they are is a single protein molecule. The cellular life forms have ribosomes. Ribosomes are the site of protein synthesis. We'll talk a little bit more about that. Viruses and prions have no ribosomes. If we look at the metabolic type of the cells, the cells produce ATP as the energetic molecule. Viruses have no ATP production. For that matter, viruses have very little metabolism. They do have some metabolism, like when the virus is replicating. But outside of a cell, they have no metabolism, and very little metabolism within the cell. Prions have no ATP production and very, very little metabolism. All right, any question about any of this? If not, let's move on to a history of microbiology. We're going to be talking about a brief history of microbiology, and the history of microbiology largely concerns the debate over whether spontaneous generation occurred or whether it did not. And the history of microbiology also encompasses the golden age of microbiology, which you don't need to know exactly when that was, but in the 1900s to the early the 19th century, meaning the 1800s to the early 1900s. And it was called the golden age of microbiology because when microbiologists were studying things in that time, everything they learned was something new. Here we're looking at the age of microbiology and just a timeline. The golden age was the start of microbiology, and I can't read that. Let me blow that up. What is before the golden age of microbiology? Oops, too far. This was what was happening in science before the discovery of microbiology. We'll talk about some of these events, the key events that happened in the history of microbiology as we're going through the lesson. All right. So when we're talking about the history of microbiology, you should understand it goes back to ancient times. The earliest record we have of A microbe is a fossil record in the rocks which is over 3.5 billion years. So over 3.5 billion years ago, we can find a fossil of the ancestors of bacteria. And they were the first life on Earth. Now, generally speaking, when I'm going to give you a history of microbiology, I'm going to be talking about it with a date. But except for this first date, you don't need to know any of the dates. Meaning I'm not going to test you like when the Babylonians wrote their first clay tablets. Okay? Any question about that? So the only date you really need to know is the first one, and that is 3.5 billion years ago. We have the first evidence in the fossils of an ancestor to a bacteria. And actually, you're looking at it here. Here is a fossil of a bacteria. That's an artist's picture of it, but there it is there. Clearly a bacteria, and it's about 3.5 billion years old. The next major event in microbiology was the Babylonians wrote on their clay tablets recipes for making beer. And they didn't know it, but the reason why they got beer was because a microbe was fermenting whatever you use to make beer, probably grain. And so this is an important event, the first writing that we know of that was discussing microbiology. Although the Babylonians didn't know it because they couldn't see the microbes. Their recipes involved making beer, which would use microbes. And then the next one was the Mayans wrote in their records, 600 AD, how to make cacao, which is a fermented beverage that they drank, which would be similar to chocolate, like chocolate milk. The difference is they didn't have milk, and they were drinking a liquid chocolate, but it wasn't chocolate milk, which is the way we're familiar with liquid chocolate. Any question about that? Now, when I ask you questions about some of these dates, you should realize that I may ask you, like, what had to happen before the Mayans could write their records for fermenting a beverage from cacao? And that is, the Babylonians had to write their records because that happened first. And then we had to have the first fossil records on the planet Earth. So I could ask you the relative order of these things. Okay? As well as I could ask you something like, gosh, what would I ask you? The Babylonians wrote clay records for making a beer recipe. Why was this involving microbiology? And that's because it was the microorganisms which made the beer. And the same with the chocolate. It was the microbes that fermented the cacao. And if you don't know, when you eat chocolate, it is fermented. So we rely on the microbes to get us our chocolate. Any question about any of that? All right, let's move on. So the next important event in the history of microbiology was the invention of the compound microscope. You should know that Janssen developed the compound microscope, but I'm not going to ask you that he invented it in 1590. And obviously a compound microscope is important for microbiology because without the microscope we can't see the microorganisms. Surprisingly, the first cells were not seen until 75 years later. Robert Hooke took a compound microscope and looked at cork from the cork tree, which is the bark of the cork tree, and he recorded his observations and found little boxes which he called cells. And let me see if I can print that out. I don't think I'll be very good at this, but I don't know how to use this program. Oh, come on. Well, I'll just pull that out of there. Come on. I'm trying to get there, I got rid of it. So, what he did was, he saw, and I'm supposed to be drawing straight lines, you can tell I'm not an artist. He saw, I'll try and go all the way down. Things like that under the microscope. And he called them cells because it reminded him of the rooms in a monastery where the monks slept and lived. And because it reminded him of the rooms of a monastery, he called them cells. The monks called them cells. And so that's where the term comes from. I don't know if that's fascinating or not, but I always like it. And they were just little boxes. He then looked at cells in plants, and he found them and recorded it. At the time, people looked at animal tissue. They did not find cells. What is in plants that would make a cell show up in a microscope in 1665, and that's your hint, but not show up the cells in an animal? What do plants have that animals lack?
A cell wall.
Yeah. Plants have a cell wall which is easily visible, and with the primitive microscope of 1665 they could see cells in plants, but they could not see the cells in animals because animals don't have a thick cell wall, they only have a cell membrane. But as the microscopes improved, they started noticing cells in animal tissue, and that marked the beginning of the cell theory. And that is, the cell theory is all living things are composed of cells. And that was developed further by others, and then formally written up by Virchow, that all living things are composed of cells. But the very beginning of it was Robert Hooke looking at plants and seeing cells. Now, the above are not specifically microbiology. The development of the microscope is not specifically microbiology. Robert Hooke's looking at plant cells. A plant is not a microbe. Okay, although you could argue a cell is a microbe, because you can't see it unless you're using a microscope. And that's not true of all cells, but most cells. All right, any question about any of that? I'm probably confusing you. All right, I mentioned that the history of microbiology largely concerns the hypotheses about how life originated. And in the 17th to the 19th centuries, meaning from the 1600s to the 1800s, there were two competing hypotheses to explain how life began. The first was that life spontaneously generated, and that was given the name spontaneous generation, where life arises from non-living things, non-living substances, and the theory was a vital force came in, and then the substances could be converted by spontaneous generation into a living organism. The other theory was biogenesis, and they said no, spontaneous generation doesn't happen. All living things arise from another living thing. And everybody knows a baby comes from their parents, and that would be the theory of biogenesis. Well, in microbiology, these two theories were competing with each other in early microbiology. Over a century ago, there were beliefs that included things like if you threw a log into the fire, a newt would come out of the fire, would crawl out of the fire. And people would say, you put the log in the fire, spontaneous generation of a newt, and it crawls out of the fire. Of course, in reality, the newt was probably in the bark of the log, and the newt felt the heat of the fire, and so that's why it crawled out of the fire. Any questions about that? Meaning, I don't believe that newts spontaneously generate from a log and fire. Another example was they noticed that flies could emerge from manure, and people would say, spontaneous generation. And in the early days, they really had no proof one way or the other about that one. And a third one was they saw maggots arising from a decaying corpse. Once again, people would say this is proof of spontaneous generation. The maggots arose from a decaying corpse. The vital forces came into the corpse, and the maggots spontaneously generated. Most of them were logical, but none of them were scientific. Some were even amusing. Like someone wrote up that if you put sweaty underwear in a jar of wheat, it spontaneously gave rise to mice. Okay? And nobody's smiling or laughing at that. What happened was the mice went into the jar to get the wheat, and then the mice couldn't climb out of the jar, and that's why they found mice in the jar. The sweaty underwear, I have no idea why that was necessary. Let's take a brief sidetrack, and that is with microorganism. They noticed that when you had a tube of media, if you left it out especially if you left it out uncovered or it's not sterile, it would become cloudy, turbid, which I don't know how well that's showing up. Let me blow that up. Get this down. Come on. That's a little better. The turbid means that it's cloudy because microorganisms are growing in it. If it's clear, there's no microbes in it. And they noticed with microbes that initially they always had, when they made their media, something growing in it. Of course, the reason is because the microbes were in the tube or in the air and could fall into the tube as long as the tube was uncaptured. And generally they weren't sterilizing the media initially and when they first did this. So of course the media became cloudy because microbes are in the tube, they'll grow and make the tube turbid, and that shows growth. The first evidence for or against the theory of spontaneous generation came from an experiment done by Francesco Redi in 1668. And you don't need to know the date, I'm just telling you when it happened. Francesco Redi is important because not only did he give the first evidence against spontaneous generation, but he also gave the first documented scientific experiment. So he wrote up the first scientific experiment. What he did was he had 3 jars completely sealed and he put meat in it. He had 3 jars completely open and he put meat in them. And he had 3 jars covered with a fine wire net. He noticed in the 3 jars that were open that the meat later on got maggots in them. But in the 3 jars that were completely sealed and the 3 jars that were covered with a fine wire net, there were no maggots in the meat. And by the way, he had the 3 jars with the fine wire net because when he sealed the jar, the people who believed in spontaneous generation said, you sealed the jar, the vital force couldn't get in the jar and get to the meat to give rise to the maggots. That was their theory for why the jars with the meat that were sealed got no maggots. So he put a wire mesh over it and you got no maggots. What happened is the maggots come from the flies coming to the open jar, landing on the meat and laying eggs in the meat, which then give rise to the maggots. And because he had the covered jar or the one covered with the wire mesh, that kept the flies out, and so there were no maggots in these 6 jars. And that was the first evidence that spontaneous generation did not occur. And after Francesco Redi's day, people came to the conclusion that complex living things, like a maggot or a person or a cat, something that's big, could not arise by spontaneous generation. However, they argued that microorganisms could arise by spontaneous generation. For example, if you covered a jar of media with the wire mesh, something would grow in that media because the microbes could come through the wire mesh. let's skip that. And let's say that about this time, the first observations of microbiology occurred. The first recordings of an observation by a microorganism was done by Antony van Leeuwenhoek. And I'm not Dutch, so I might be butchering his name, but I think I'm pretty close on pronouncing it. And he described seeing microorganisms in almost everything he looked at. He looked at microorganisms from scrapings from his teeth. He looked at microorganisms growing in rainwater. He looked at microorganisms growing in a peppercorn infusion. And he looked at microorganisms that he found in the River Thames. If you don't know, the River Thames was flowing through London, and apparently the Londonites drank that water to get their drinking water, and they weren't too happy to hear that there were microorganisms in the water that they were drinking. The reason why Antony van Leeuwenhoek was the first person to observe a microorganism is because he was the best microscope builder of his day. And here we're looking at one of his microscopes. It's a simple microscope, meaning it only has one lens. And the reason why his microscopes were the best was because he was the best lens grinder of his day. He actually ground his own lenses and then built his own microscopes and then looked at organisms, including microbes. He may have also had very good eyesight. Another thing that Antony van Leeuwenhoek is famous for is this drawing of his here. If you don't recognize it, all of these cells are bacteria. We haven't talked about bacillus, but this is a bacillus, that's a bacillus. These are cocci. This is bacillus, that's a bent bacillus. And that is a spiral. We'll talk about those in a little bit. Those are different type of bacteria. These are cells so small that no one else saw them for almost a century. The reason was that his microscopes were so good he could see these very small cells, which were bacteria. The other microscope of his days were not good enough to see bacteria. And it took 100 years of improvement, of different people improving on the microscope, before the microscope got good enough so that other people could see bacteria. So he saw bacteria almost 100 years before everybody else. Any question about any of that? If not, I think we're out of time to stop the lesson, so let me take a look here. If anybody knows, do we stop at 3:20? Nope, 3:45, so we have a little bit longer. Sorry for getting your hopes up. We'll go a little bit longer, but I'll probably end a little early. Rudolf Virchow wrote up the theory that all living cells arise from pre-existing living cells, which was known as the concept of biogenesis. This theory had been around before he wrote it up, but he formalized it and wrote it up in 1858. However, at this time, people were still arguing that microorganisms could arise by spontaneous generation, and who finally ended the belief that microorganisms could spontaneously generate was Louis Pasteur. In 1861, you don't need to know the year, he published results showing that spontaneous generation did not happen. So Louis Pasteur disproved spontaneous generation. And this is someone you should get to know, because we will talk about him several times in this course. Louis Pasteur was one of the founding microbiologists, and we will run into him time and time again in this class. Anyways, he disproved spontaneous generation and therefore confirmed the theory of biogenesis. What he did was he demonstrated that microorganisms are present in the air and that microorganisms can be destroyed by heat. And he made an experiment where he had an ingenious and famous swan neck flask where he destroyed the microorganisms by heat and then prevented them from the air from getting in, but kept it open to the outside so the vital forces could come in and cause spontaneous generation. And guess what? They did not. What he did was he had a glass flask like this. He poured in his media, and then he heated the glass flask right here, bending it into a swan neck. This is important. So you can see that, well, let me say, he then sterilized the flask by heating it, killing the microbes which are in the flask and in the glass. And then he left it open to the outside so the vital forces could come in, but they didn't. The media stayed non-turbid, there's no growth there. What happened is the microbes from the air could come into the flask, but they end up right here. By gravity, they stop right there. They do not go up the glass flask and then into the media. If, on the other hand, he pours the media down into this section of the flask and then bring it back, he could get growth, showing that the growth that happened in something like this, that's left open to the air, happened because of microbes in the air. And so there's no microbial growth in this, meaning spontaneous generation did not happen. And the theory of spontaneous generation then was thrown out the window, and now we only believe in the theory of biogenesis. Any question about any of that? All right, well, beginning with Pasteur's work, other discoveries were made, and that began the golden age of microbiology, where discoveries were made like the relationship between microbes and disease, and that gave rise to the germ theory of disease, and this is that germs cause disease. It also gave rise to immunity and gave rise to the concept of antimicrobial drugs. That you could use a drug to prevent or cure a disease. Any question about any of that? Some events that were important in the early age of microbiology, which were part of the golden age. In 1850s, you don't need to know the date, Pasteur developed the basic idea of aseptic technique, where you have a culture, which in this case, there's nothing growing in it, but if you were to put one cell in it and then it were to grow, it would be aseptic because things from the air can't fall in and then contaminate it. Any question about that? In 1881, Koch developed a technique for streaking for isolation on solid media, and Louis Pasteur did develop a technique for getting a pure culture. A pure culture is one that only has one species in it, but Louis Pasteur's technique was really difficult and time-consuming. And Dr. Koch developed a simple technique for getting a pure culture, and that is streak it on a solid media. You do it and get isolated colonies, and you take from the first isolated colony and you streak it a second time and get an isolated colony. If you take from a colony that's gone through 2 colony isolation events, you most likely, greater than 95%, have a pure culture. Okay? So Koch developed a simple technique for streaking for isolation, allowing us to easily get a pure culture. In 1882, Hess, a technician of Dr. Koch, developed nutrient agar, a very simple media that allowed for a large number of microbes to be grown. And then about the same time, in the 1880s to 1892, Winogradsky developed the idea that microorganisms are involved in the biogeochemical cycles, such as the nitrogen cycle, which we discussed in our first lesson. But he also studied the sulfur cycle, where sulfur moves around in the ecosystem because of different microorganisms. And like I said, it's curious to get his biogeochemical cycles. Winogradsky had to study a mixture of many different types of microbes. And he was different from the other early microbiologists like Koch and Pasteur. They were working with one species, a pure culture, and then studying that pure culture. Any question about any of that? All right. I think this is a good place to stop, and I promised I'd stop a little bit early. The next couple of slides will take more than 2 minutes, and I wanted to stop at 3:30. So I'm going to stop here and ask, are there any questions? If not, I will see you at 1 o'clock tomorrow. All right.
Okay.
Oh, go ahead. You got a question? You got to unmute yourself.
I have if I do the lab today, if I didn't do it yesterday, like what does that technically do?
Okay, can you get a little closer to your computer? I'm turning up the volume. I couldn't hear all of that.
Since I wasn't able to complete lab 1 yesterday, can I complete it tonight for credit?
Yes, you have until Saturday to turn
Oh,
in the worksheet for, it's not lab 1, it's lab 00. We're going to
okay.
do lab 1 tomorrow.
All right, thank you.
Okay. All right, if there's no further questions, we're going to end here. And I'll see you tomorrow.